What does our DNA code for? Well, this is interesting. So humans have about 3.3 billion base pairs in their nuclear DNA. And obviously that includes genes. How many genes? Well, that has been debated for a long time. So for example, when I was in school and when I first started teaching, the estimates of the number of human genes was about 100,000. And I think most people felt rather comfortable with that because, if fruit flies have, say, 17,000 genes, obviously I'm much more complex than a fruit fly. I must have a lot more. So 100,000, that was something that we were comfortable with. However, it turns out that was a large overestimate. And the reason that I say that is because now it's appreciated in a way that it was not before, that one of the things that gets, one of the ways that RNA is processed after transcription is that introns are removed. And so the DNA sequence is copied, but there are a number of intervening sequences which then never become protein. They get subtracted, and then the remaining parts known as exons get pasted together. All right. But it turns out that perhaps one of the major reasons for introns is that this gives the different cells of the body the chance to splice the exons together differently. Just when the introns are taken out, if each of these colors represents, a protein portion which would be made by a specific exon, a cell could decide to put all of these exons in the protein product, but they don't have to. So a liver cell could perhaps use some of these exon sections, and a nerve cell could use other sections. That would produce different proteins that they would share perhaps some of the same sections, but then they would differ. They would have different sizes, etc. And early on, this caused an overestimate of the number of human genes. What wasn't appreciated is that one gene could be spliced together differently to produce these alternate versions, which could be recognized as being distinct. And so the early estimates went from, say, 100,000 to 80,000 to 60,000. And I'm speaking of this lightly. I'll be perfectly honest. It was infuriating for someone who then was trying to teach genetics, because then all throughout my notes, I would have statements that, oh, there are 100,000 genes in the human genome, 80,000, 30,000, 20,000, depending on what year I had written that. And then, very often I'd have then multiple statements that had to be updated, et cetera. So the current estimate is maybe, say, 20 to 25,000 protein coding genes. But as I'll pursue again later, this gets so much more confusing. So I just want to state this. But let's say, all right, here's all our DNA. Let's say that this is 100% of the nuclear DNA, with each of these red DNA double helixes representing 1% of the genome. The genome is the DNA organized into these 46 chromosomes from the nucleus. Well, not all of this is transcribed. A lot of it is, but not all of it. How much? Well, that varies from cell to cell. But if you were to try to come up with, an organism-wide average, maybe you'd come up with 70%, 80% is what's called the transcriptome. So the genome, all the DNA, is different from the transcriptome, which is transcribed. Now, I think a lot of people would assume, oh, these are then protein coding genes, correct? No, only about 3% of them is. And so 97% of the genome is not converted, does not code for the amino acid order of proteins. So the proteome, that portion of the genome which encodes that amino acid order, that's actually, once again, only about 3%, actually quite small. But then the question comes up, and this I'll get back to, what what do these transcribed portions of RNA do if they are not coding for protein? If we have 70% of the genome is transcribed and only 3% is translated, what is the rest of this doing? Well, a lot of it has functions, which I'll get into, but then that can raise the question as to what a gene is. So obviously the transcribed RNA, which then becomes protein, these are genes. But what are these RNA molecules doing? What percentage of the genes never become protein? This is in a bit of flux. And since some of these RNA molecules can be small, it's quite possible that they're hard to identify and that we miss some of them. So there's that component. Now, it doesn't matter. And so we're in the process of learning these things, and it doesn't matter in the end. However, once again, as I said, when I started teaching, when I was in school, there was certainly the idea that more was better. All right, that we would have a lot more genes than fruit flies, because we're so much smarter than they are and more complex. So we would have a lot more genes, and even then like more chromosomes. They have 4, we have 46, but you can't correlate the amount of DNA or the number of chromosomes with complexity. And so it's not, numbers don't mean anything. So for example, if we look at the numbers of DNA, dogs have 78 chromosomes while we have 46. Horses have 64. Carp have 104 as a diploid count. There are ferns which have more than 200. And so clearly, there's not a correlation between the complexity of an organism and the number of its chromosomes. More chromosomes don't make you better or smarter. In the same way, more DNA doesn't make you better or smarter. In fact, after a certain point, it would just make you less efficient. If you had extra DNA that you didn't use, it was just now more costly for every cell division. So if you look at yeast, Drosophila chickens, and humans, and ask how many picograms do you have in a haploid set, humans have 3.2. We're higher than those other ones that I used. You might argue, we're better, we have more DNA. But if you were to take wheat, beans, and onions, they have far more DNA per cell than we do. There's a type of algae known as a dinoflagellates, a protist. And they're, just weird in so many ways. But if a typical alga has half a picogram of DNA per cell, dinoflagellates can vary from 3 to 215 picograms per cell. Once again, 3 is comparable to humans. 215, obviously that's so much more bigger than ours. We have 3 billion nucleotides, but dinoflagellates can vary from about that size to 200 billion nucleotides. Once again, that doesn't correlate to better or worse, more complex. If anything, if you had extra DNA, that would just make it more costly to perform DNA replication. More after a while would be wasteful. So we have all of this DNA. What's specifically in the sequences? So obviously, genes are encoded in our DNA. So let's imagine here there's a yellow gene which encodes a specific protein. Some of these exist as a single copy. There is one copy of this gene in the genome on a specific chromosome. However, one of the greatest conclusions of the Human Genome Project and the study of genomes is the role of gene duplication. So what sometimes happens is that when crossing over occurs in meiosis, it's sloppy and a mistake is made so that one of the sister chromatids ends up missing that gene and the other one now has 2. So on this pink chromosome, that gene has been duplicated. Where there was once 1 copy, there is now 2. Now, if the 2 sequences stay the same or essentially the same, now you just have 2 of the same protein, or the same gene coding for the same protein. So now there are multiple genes in different spots of the genome, perhaps because this can move ultimately, which code for the same protein. So the gene exists in multiple copies. But let's say there's a mutation here that changes the 5th guanine to a cytosine. And then later there's a mutation here that changes the 1000th thymine to an adenine. Well, after a certain point now, these are no longer the same gene coding for the same protein, but now instead they are coding for different genes in a gene family. One of the greatest conclusions of the human genome is that our genes often exist in gene families. And so sometimes this can be dispersed throughout the genome, or it could actually be in a tandem array, like all on the same chromosome in a group where there's a whole bunch of them. You could get a series of genes which came from one original sequence, but then since then have created separate genes. On my YouTube channel, I have lots and lots of videos on this, so I'm not going to go through this to a great extent now. But there are so many gene families. So a kinase is an enzyme that adds a phosphate to a protein. A phosphatase removes it. Do we have these? Yes, we do, but we don't have just one gene. We have hundreds of kinase genes, hundreds of phosphatase genes. They're modified duplicates of the same ancestral protein. So here was a functional protein, and then at some point there was an extra copy and another extra copy, et cetera. They changed, and so now they all do the same thing in adding a phosphate to a protein, but they're doing it in different ways. And so now some can be involved in cell division, some can be involved in differentiation, et cetera. We have over 1,000 G protein coupled receptors. So these are receptors which bind something on the outside and then activate a change on the inside of a cell. That's a great system to do that. But the fact that we have 1,000 of them indicate that some of these can be interacting with light. Others are opposite. Some of these are interacting with olfactory odorants. Some of these are reacting with things that we are tasting. Some of these are reacting with hormones. And so when you ask, do we have a receptor for the hormone prolactin? Yes, we do. Do we have a hormone, a receptor or a gene for the receptor for the hormone GnRH? We do. But they are all G protein coupled receptors. They are all members of the same gene family. Once again, we have about 1,000 of them, with hundreds being olfactory receptors. And so therefore, if you're trying to study the genes which make up the human genome, you don't have to remember 21,000 separate genes. There are these gene families. There are hundreds of transcription factors in different families, like the zinc finger transcription factor family, or the helix loop helix, or the helix turn helix. So we have lots of ion channel genes, et cetera. And so one of the things that we see is that gene duplication has produced lots of separate genes which can now, which have varied over time and are different genes in the same gene family. Now, sometimes they stay pretty much the same. And so if you look at translation, for example, we know that for translation, you need tRNAs specific to each of the amino acids. We know that you need lots of ribosomes. They're proteins, there are RNAs. And we often find that then the tRNA genes are present in multiple copies. So two things to focus on here. Both the tRNA in this video and the rRNA in the next one, they never become protein. So what the DNA codes for this, but this is the final product, this RNA molecule. It does not become protein. And so a lot of our genes make functional RNAs, which aren't protein in the end. And important ones like the tRNAs, we can have hundreds of copies of the same tRNA, because a cell that's actively making protein needs to have enough tRNA to bring with specific amino acids to all of the ribosomes of the cell. One gene might not be enough. And so cells often need multiple copies of the genes for tRNA, multiple copies of the rRNA genes. So if you remember the ribosomes where proteins are made, they are complexes made of lots of little proteins and then molecules of rRNA. Well, because cells need lots of ribosomes, making proteins is a major function of the cell, then there can be lots and lots of genes of these individual proteins or the rRNA subunits, etc. And so thinking of genes, then is a bit more complicated because once again, there are genes which code functional RNAs that do not become proteins. And so this would include the small nuclear RNAs, which are used to make the spliceosomes for splicing genes. And so notice here we have a different, so here we have a different genes which we need, but they make RNAs which never become protein. Now, these spliceosomes are also essential. So very often, one single gene with a specific sequence has multiple copies, very often all existing in a row, what's called tandem arrays. And so these are proteins, as are so many other. And once again, because so much of the so much of the genome is transcribed, but not translated, then there are then apparently a lot of different there are a lot of different functional RNAs which never become protein, and so that can be a topic for another conversation, but there are small nuclear RNAs, small nucleolar RNAs. There are RNAs which turn off X chromosomes that are involved in gene regulation. Here's another class which is odd. They're called microRNAs. They're only about 21 nucleotides long. And I emphasize that because you see how small they are? Obviously, as a result, you could miss them, all right, because they have a function, but very often when you're looking for genes, you're looking for certainly, typical or canonical sequences where you say, a lot of genes have this sequence. Let's look for such things. But if these functional RNAs are small, then it would be possible that you would not see them. And so just before I get into the really weird sections, what is then a gene? Well, a lot of them are coding for RNA molecules that never become a protein, and many of these are present then in multiple in multiple copies. And then there are the weirder sections, which are relatively newly discovered and are exciting because these are areas of active research as we try to understand how cells work. The experiments which discovered them were those called DNA hybridization. So if you heat up DNA, you separate it into single strands. And then if you take specific sequences and you ask, how long does it take for the sequences to rejoin? If a sequence is only present once in the genome, it's going to take a little bit of time before that sequence can find its pair. So it will take a while before that becomes double-stranded again on average. But if something exists as duplicates, if there's extra copies of a certain sequence, then it won't take long at all for that sequence to become, to find a partner and to become double-stranded once again. And when you study DNA hybridization, you realize that there's a lot of DNA which must have millions of copies because these sequences, they can be maybe short, or 100 um, base pairs in length, 5 to 10 base pairs in length, that they come together so quickly that they must be present in a great number of sites. Some are highly repetitive, as some have fewer copies and are intermediately repetitive sequences. This includes the transposons and the retrotransposons, which I will be talking about presently. Some of these are referred to as lines and some as signs, these short interspersed elements. And the major one is recognized by this one enzyme that cuts DNA, an endonuclease called ALU. And so these are called ALU sequences. So there are short interspersed sequences, there are lines, long interspersed sequences, and there are endogenous retroviruses. And so, then the point is there is a lot of unexpected sequences, in addition to those, say, coding for protein, et cetera, many of which are highly repetitive. What are these sequences doing? Well, I'm going to go through it, and there is a little bit of an overlap. So, for example, talking about transposons first is needed before talking about, the ALU sequences afterwards. So, here's a piece of DNA. It's called a transposon, and you'll see why in just a second. It codes for a protein, all right? So it codes for a protein called transposase. Transposase then can cut this DNA from the site where it was located in the genome, transport it and insert it into a different place. This is what's called a jumping gene or a mobile element. This DNA used to be over here, maybe on chromosome 5, but now you wake up one day and now that piece of DNA is somewhere else on chromosome 10. It moves. So a transposon is a mobile genetic element which can actually move throughout the genome. Now, moving about the genome can have some consequences because where it inserts, it could actually interrupt a gene, break a gene from its regulatory elements, and could end up being a cause of disease. And this is known. There are diseases which are caused by transposable elements moving. Some of these have lost their ability to move because of mutations which happen over time. Some of these can move using the transposase encoded by a different transposon because theirs is no longer functional. But some are, quote, hot, capable of transposition. And all of us have these transposons which are hot, and potentially they can move throughout the genome, perhaps causing disruptions when they do. So these jumping genes are important. Another group of elements to consider are retrotransposons or retroposons. So these retrotransposons, they go from the RNA form back into the DNA. So we know that DNA can be transcribed and an mRNA is made. And you can tell the difference between an mRNA sequence and DNA sequence because mRNAs, they have the introns removed. All right. So you'd think that once we make the mRNA, the introns are removed, we have a poly A tail, or we can recognize mRNAs. But instead of now being translated outside of the nucleus, it could be reinserted as DNA. All right. And then when the cell divides next, it will replicate a DNA copy of this. And so you can tell that this was once an mRNA because once again, it's a duplicate of a DNA section, but it's missing the introns and it has a poly A tail, all of these adenines at the end. And so you can see this was an mRNA, which then was reinserted into the genome. And so now something exists as multiple copies. Now, the interesting thing is if you wanted to follow the evolution of these, some of these jumping genes, some of these lines and signs, you can say, this arose in ancestral amniotes and only the amniotes have it. Or this arose in ancestral primates and only primates have it. So these mobile elements have jumped into genomes at different points in evolutionary history, and therefore they affect some lineages more than others. Or so SVA elements are affecting primates, while P elements are affecting, say, fruit flies. And so there's that interesting aspect to them. Now, one of the tough things that I struggle with is I certainly want to say that these are banned. And that was originally, I think, the interpretation of them. Jumping genes, retrotransposons, bad, bad. You can cause mutations that can, cause disruption. So that's certainly true. Mutations in me could end up causing cancer, or mutations in the germline could cause a child to be born with a genetic defect. So those mutations can be bad. But mutations in general are simply changes. And obviously, it would be hypocritical of me to say anything too negative about changing the DNA, because I am not a normal old ape. I am a modified ape, which we call a human. Right? Apes are not regular old primates. They are modified primates. Mammals are not regular reptiles. They are modified reptiles. So mutations have gotten us to where we are. If there was no mutations, there would only be simple reptiles descended from the first reptiles. There would only be simple mammals descended from the first mammals. There would only be simple primates and simple apes descended from those first lineages. We humans would not be here. And when we look at what makes our genome different from, say, that of a chimp or a gorilla, a lot of the differences are in movements of these mobile elements. Now, some of them, as they move, they change genes. They change levels of gene expression. Maybe they cause mutations. Maybe they inactivated genes. So even though I'm not the hugest fan of mutations, nevertheless, I wouldn't be here if it wasn't for mutations, many of which have helped to make our lineage different from those of other lineages. That even goes with viruses. Right? Because viruses can insert themselves into their host genome. It can happen in bacteria, it can happen in humans. So things like herpes viruses or HIV can actually insert themselves into the human genome. Now, if that happens in your cheek lining, or if that happens in T cells in your blood, it obviously doesn't affect your children, because blood cells and cheek cells, they don't have a role in making babies. But if these viruses inserted themselves into the germline, the cells of the ovaries and testes, which are going to produce gametes, now the next generation in their human DNA receives viral DNA as a small component of it. And when we search the human genome, there are endogenous retroviruses in our genome. We have a lot of sequences, maybe 1% of our genome did not originate as human DNA. It originated as viruses that our ancestors got. Perhaps some in the ancestors of primates, or some in the ancestors of amniotes, etc. Now it's human DNA now. All right. And so just because it originated as a virus doesn't mean that it is, it's part of our genetic structure now. And because many of these sequences disrupted genes or caused mutations, some of these viruses had a role in making us human. All right. Because once again, if there were new mutations, we would still be apes or reptiles or fish or whatever. And so even though I'm not a fan of mutations per se, and I'm not a fan of changing the genome. Nevertheless, I wouldn't be here, none of us would, if the genome wasn't changed from our ancestors. And so there are all of these different ways of changing the genomes. And let me say one word about the ALU sequences, because we have so many of these signs, these short interspersed elements. They started apparently in primates, and they have expanded greatly within primates, especially in the apes, and certainly in our lineage. Excuse me. Um, and so we have, just a million copies of these ALU sequences. What is their situation? Well, they encode an RNA, and the RNA then goes to be translated. But if the cell also has a transposon, the proteins made by the transposon can be hijacked, if you will, by the RNA of this ALU sequence. And then the transposon can then reinsert the ALU sequence into the DNA, thus amplifying it. So there was a DNA portion of it that was copied into RNA, but then this RNA then gets inserted somewhere else in the genome. And so we have, so these ALU sequences, they use the machinery of lines. So these are short interspersed elements, but they're using the line machinery in order to spread throughout the genome. We have more than 1 million of these sequences. And while they can cause diseases, because when they jump into a new part of the genome, then they obviously disrupt it. But then, like I said, I want to say bad, 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 but they differ between us and chimps. So once upon a time, chimpanzees and humans had the same ancestors, but then there were changes in each lineage. Some of these changes included the ALU sequences where they were inserted. So these jumping genes and the changes that they've made, they've helped to make us who we are. And there are different families of these jumping genes, which then have had different histories. So for example, in fruit flies, there are transposal elements known as P elements. There's another, and these P elements, these P elements can make the difference between, say, two species like Drosophila, melanogaster, and simulans try to hybridize. The embryos might die. And one reason might be if one of the species has mechanisms for controlling the transposable element, and the other species doesn't, now their hybrids might die because of then a dysregulation of that. Okay. So when we look at human sequences, as I had said, there's what we expect. There are the sequences of genes which code for the amino acid sequence in a protein. But then there's so many other things, like we find that many genes are present in multiple copies, that modified duplicates form these gene families where we can have hundreds of kinase genes, hundreds of GPCRs, hundreds of zinc finger protein receptors. I'm sorry, zinc finger transcription factors. But then we realize that there's so much RNA which never becomes protein. The RNA is the final product. So therefore our transcriptome is bigger than our proteome. And then we see that there are these mobile elements, these transposons, retrotransposons, these endogenous viruses which have inserted themselves, and diverse classes of these mobile elements, signs, lines, that are moving around, significant in the medical field because they can be a cause of disease, but also significant in an evolutionary sense because these have helped, they've contributed to the changes in genomes, which then result in the evolution of life on Earth. So the study of sequences leads to a large number of very interesting observations.